Hello everyone and welcome to this Certara webinar on how to save valuable time on PKPD analysis and modeling using Certara's Phoenix software. My name is Marie Bonnard and I am the EMEA zone leader at Certara Software, and it is my pleasure to introduce today's speaker, Simon Davis. Simon joined Certara in 2005 and has been supporting and training scientists on Phoenix and Integral. He is also a regular speaker at Certara webinars, and you might have joined last week's webinar on Integral, but also at other events such as PK UK and AGA. Without further ado, I'd like to introduce Simon.
Thank you for that introduction, Marie. Sebastian, were we going to put a quick poll up just to get a measure of people's background? This is an anonymous poll, but it's just to see if there's anything in particular I should focus on in the presentation. Thank you, Sebastian. So we'll just give you another 15 seconds or so. It's quite easy, no trick question there. Okay. So and are you presenting the results now, Sebastian?
Not yet, actually. Yes,
Okay.
we are now seeing them.
All right. Okay, so mainly NCA users. Okay, great. I will move on. So as is usual with these things, we have all of you muted at the moment. If there's any questions, please ask them in the Q&A panel, and a recording of today's webinar will be provided shortly, probably go up tomorrow on YouTube. So I'm just going to turn off my camera, so I find it a bit distracting seeing my face while I'm talking, and we will start. So what do I want to cover today? Phoenix, as you may know, is more than just NCA. It is a platform for not just non-compartmental analysis, but also for compartmental analysis, whether that's individual or population analysis. It has other tools like bioequivalence. I'm sure some of you are using those. And we've done many presentations already about templating, using bioequivalence, relative scaled average bioequivalence, and those webinars are all available either through Certara University or YouTube. And similarly, we've got other material on IVIVC for formulation development, population effect modeling is nonlinear mixed effects, and that's going to be the main focus for today. We also provide validation suites to help accelerate your validation and let you use and get value from the software quicker and faster. There are additional plugins to support CDISC and automation of workflows. These are PK Submit and the PK Assist plugins. And of course, as Marie mentioned, there's the Integral repository, which supports Part 11 compliance. It's hosted software as a service, so that means you can get up and running very quickly. And there's a recorded presentation from last week if you want to find out more about that. What I want to talk about today is what your pharmacometricians are doing and what you as NCA scientists can maybe get more value from Phoenix and learn more about your compound and accelerate your drug development. So population PK guidance from the FDA has been around a long time. I think their first published one was in 1999. They recently updated it in February of this year. And why do we do population modeling? In fact, why do we do modeling at all? Non-compartmental analysis is very much based on measuring from a defined protocol, and it's chosen because it's not got a lot of room for interpretation. It's a very definite. We're measuring the shape, the height, the area of those curves. With population modeling, we are trying to build models which can predict how the PK might change with different dosing regimens, maybe loading doses, different dosing frequency. It's much more powerful tool because we've got a model where we know the parameters and therefore we can extrapolate for different data. We can add in the exposure response, and this is really the key point of drug development. We're not interested in the Cmax, so to speak, of a patient. We're interested in the maximum effect and the safe effect of that drug. So we need to build up an exposure response profile, and if we do that successfully, we can even use that to approve dosing regimens which haven't been actually studied in the clinic, and we can propose new dosing regimens from that. Coming back to your use in NCA, you can use modeling to better define your sampling scheme requirements and also sample size calculations. But going on, if we've got these exposure metrics and we can build a model where we've got relatively low residual error between subject variability, part of what we do to get there is we bring in covariates. So these covariates are characteristics of the patient, whether they're got particular disease states, age, body weight, sex, etc. And we see how those alter the drug's behavior, and we characterize and most importantly quantify that. And when we've got that built into our model, we can do better drug labeling, we can measure and predict DDIs better, we've got better informed pediatric labels or pediatric trial designs. So I mentioned this paper from paper guidance from 2022, and it's really useful, I think, if you've not done a lot of population modeling before, or in fact a lot of modeling, is it helps you think about what steps you do. And it's about 25, 27 pages long, but it's worth reading. But I'm going to summarize a few points of it here. First of all, do your examination of data. That might be tables, quite often going to use the plots. We then go into the model development. So model development is it one compartment? Is it two compartments? Have I got linear or non-linear clearance? And then we go further to try and explain that inter-subject variability. And one way we do that is try and identify covariates, as I just mentioned. And we can do that in a statistical way. We can search, it's called a stepwise search, try adding in different covariates. And I'll show you how that works in the demonstration later. And that's one of the recommended methods from the FDA, stepwise covariate search, and we provide that in Phoenix. How do we handle missing data? How can we interpolate between those values? We've got the model validation. Again, there are tools like goodness of fit plots, visual predictive check, bootstrap. All of these methods and techniques are built into Phoenix to be accessed as a click. And the reason I'm mentioning this is I feel that with the Phoenix interface, which you're familiar with from NCA, you're comfortable with that sort of user interface, graphical user interface, but you can spend more time learning the science and the background and less time programming around with codes to try and make various different tools work with each other. It's all there in one package. Going on, turning a fitted model into a simulation is very simple, and you can build up different sorts of simulation based on the uncertainty of those fixed effects estimates and that between subject variability in potentially new populations as well. And the guidance goes on to talk about how population modeling is suitable for all stages of development, whether that's your new drug applications all the way through to post-marketing, label development, new populations, new uses. Most importantly, when you go into modeling, you have to still consider that although there's the mathematical approach, and you can find a mathematical model that fits anything, is it still biologically plausible? Does it match up with what you know about your compound and biology already? And are those parameters identifiable? Do you have confidence in there? There's no point in having a super complicated model, but you can't actually predict anything with confidence. So we do this in a variety of ways, but essentially we do our early stage clinical trials Initially, you've got safety trials in phase 1, efficacy comes in as your phase 2A studies, and later on phase 3, you're concentrating more on dose determination. And during this time, you're predicting those drug concentrations, and most importantly, the responses in that intended population. And then later on, as you've quantified the variability in those relatively homogeneous populations, you can go on to maybe firstly predict an effective dose regimen. So you know within a confidence interval, typically 95%, that no more than say 20% of the population will reach any toxicity with that patient. And then you maybe have got suggestions for how to modify and monitor that TDM, it's called therapeutic drug monitoring, to manage those dose levels afterwards. And population modeling, importantly, allows us to make these quantitative assessments and these predictive assessments. Okay. So really it's about describing the variability, what's due to within the subject variability and what's due to the between subject variability. And it's the between subject variability that is most interesting, because you can explain that, as I said, with these covariates. It's very powerful because you can use it with very sparse data, which you might not be able to build an individual model on. And most importantly, it's this determining of covariate relationships. How does Phoenix help us do what the FDA and other regulatory authorities suggest? It's got a very, hopefully, friendly, intuitive graphical interface to work with. You can pick up these built-in models. From the built-in library, you can select literally hundreds of different variations, and then going into the graphical model builder, you can make even more complexity. And at any point, you'll see the text of that model. And if you want to edit that code directly, there is a textual editor as well. So it's a very easy way to learn how that code works and to build on that going forward. We've got a powerful engine. The Phoenix engine you're already using if you're doing individual modeling, that's the naive pooled method, but we can take that further using the standard sort of FOCE, first order conditional estimation methods, or we've got newer quasi-random expected maximization methods as well. So this is a powerful tool, easy to learn, but also supporting the latest science. We've got nice friendly tools like an initial estimates tab, which I'll tap on later and show you that. And the Phoenix modeling language is common to all of these. And for those of you who are interested in taking this further, some groups like to work in code environments and batch up jobs, run them from R. And that same Phoenix modeling language is at the heart of RSNLME, which some of you may have seen about in some of the other presentations from Certara over the last few months. What do I mean by graphical model builder? Exactly this. If you can picture what your model looks like, you can probably build it here in Phoenix. You've got options to add different compartments. This is the green box is an observation. You can have different sorts of clearance, nonlinear. You can see options here to add in T-lag, saturating clearance, whether it's infusions possible. You can freeze any element, etc. Lots of options for different residual errors. And we can build this up. And all the time you're seeing the full statements, and a more detailed text is available here later. I'll show you that. This is the initial estimates tab. This is a very simple model here, just one compartment, volume and clearance. You can see a scroll wheel, and you can see observed data, those are the circles, and what does the predicted curve look like through that data. This is an example of a naive pool, so we're going to treat all the subjects as if they were the same here. And that's why there's just one predicted line. So I've got a couple of slides here. They're not there to scare you. It's just some of the maths. When we talk about these terms, you'll see a table later on in the demonstration called the theta table. These are the fixed effects. These are the parameters you might be used to, the final parameters like you see in NCA, and these are the structural parts of the model. How do we think that drug is absorbed, distributed, metabolized, excreted in our organism? And then the random effects, this is how we define and most importantly quantify the inter-individual variability. And this is called eta. So the guys who developed population theory back in the 70s liked all these Greek terms. They come from a statistical background. So some of it's learning new terminology, but hopefully it's pretty friendly to move forward here. So what our aim is to calculate the clearance in any individual. You see this I, that will be the typical value. This is the theta symbol for clearance, plus the adjustment for that individual, which is this eta symbol here. And this example here is that we typically expect etas are normally distributed. So in this case, we've got a log norm distribution. And the reason is that we want to have clearance always be positive, right? We don't want to have a negative clearance calculated, and that's the sort of typical distribution you'll see, but you can choose other distributions very easily within Phoenix. And again, don't get worried too much about these formulas. The software very much helps you with that. And when we talk about residual errors, what this means is at any point in time, so for an individual, you have an observed concentration for an individual I at time J, and then we predict that concentration for subject I at time J with this residual error. So this is the error that's unexplained by any other covariate effects we've built into the model, the between subject variabilities, the remaining or residual error. And why have I put up those couple of pages of long equations? Well, because I wanted to show you what that means. Essentially, it's there to answer the question, should a drug be dosed differently according to some covariate? Here's an example of age. So we've got volume plotted against age. And you can see here there's a distinct trend in the data. This is the eta. This is the difference from the typical value for an individual as to what we are supposed for an actual specific individual. And you can see there's a trend by age of that subject. So then we think about, well, is this a significant trend? And is it clinically relevant? And do we need to build a model that takes account of that? And if we do build a model, do we get more precise and more useful predictions in the future? And one of the ways we do that is this covariate search stepwise. There's lots of other resources. I just picked out half a dozen here. There's a link to the final guidance just from February this year. There's a couple of short tutorials if you haven't seen it all before. What's new in Phoenix 8.3, the NLME 8.3 tutorial, which is accessible through the help, introduction to population modeling. So these are Certara University courses. Some of these are free. The first couple are. This one's a paid course. And then we've also got various tips. All of these below here are free for the support forums. If you're not using those already, I think they're very useful. And I did mention RSNLME that uses the same language, so you can go to that resource and see a whole library of built-in models are available there as well. So I'm going to move to the software itself. So I'm just going to tab through to Phoenix. I've got this open already, and I want to take you through the environment briefly. I think most of you have seen this before. So this is where you'd load projects. You've got options to insert your NCA. Most of you seem to be using it already. What we're doing here is inserting maximum likelihood models. And if I take this example here, we can see a set of data. And just like you'd always do, right click, send to modeling, maximum likelihood model. That model gets inserted. And then you can start to build up how that model might look. So at the moment, you can see it's IV. And if I look at the graphical representation, we've got a dose point directly to the central compartment. As soon as I change that to extravascular, then that extra absorption process is added to the model. And I can say, okay, I think I'm going to be better suited by 2 compartments. And you can keep building those up. You can change the residual error and so on. All of these, as we change them, you can see the statements being updated here. I can see the model text behind that. I can change the parameters here from closed form to ordinary differential equations. We can choose different parameterizations here. We can move on to edit as graphical. Say yes. And then all of this becomes interactive. I might have some other observation I want to add in. I can drag that in. So we can make these flow to be, say, one way, all sorts of things. And ultimately I can move into edit as textual. And I've got some errors here. The software helps me find those. You can see because I've defined COPS twice. And if I start to get rid of those, you'll see the errors change. So like I say, I've got MOPS, for instance, and that error would disappear. So that would be my metabolite, for instance. Similarly, if I put in some option in the toast, it's saying, okay, you've got a dose point, but you're trying to qualify it by something. And what are you expecting? A T lag, a duration of infusion, a rate of infusion, bioavailability. And these are expected terms you might use to qualify your dose point. You can see that's fairly accessible, even if you have not written code before. And we've got a lot of support for that. If I go to the help menu, click on the contents, you've got all of the stuff you should be used to. The tutorials, you know, these for the NLME, IVIC, Win and Lin. These are direct links to little videos. Obviously got the release notes up here. Down here, we've got the maximum likelihood models, how those work, graphical model interface. But what I really like working through is the different examples here. So I click on the maximum likelihood examples. You can see we've got different ways of building models up, fitting PK and PD data and using simulation. Maybe you want to create a 2 dose point model. You want to fit IV and oral data simultaneously. How to build up covariate modeling. All of these are linked. And if you go to the installation folder, I'm just flicking to that now, you can see this examples folder is full of the completed projects, but also the supporting files to build those up, source data sets, models, etc. So very well supported with the base software, its documentation, the forums, which are freely accessible, and webinars such as this, and additionally also the Certara University, the training site. So if you want to move into this area, want to do the latest science, this is a very easy environment to do this on. Let's move into the last sort of 10 minutes of the section, an example of a population model and why we want to do this. So I'm looking at the set phenobarbital. Let's have a look at the data very quickly. Here we see that's one individual, and it's only got 2 actual observed concentrations. It's got a loading dose here at 25 and then a maintenance dose of 3.5. These are neonates, premature, very small body weights. You see just 1.4 kilograms. And we've also got some response data. In this case, it's the APGAR score. This is a measure of how healthy that individual is. In this case, it's just a screening value. So we're going to be treating it as a covariate in this exercise. So you can imagine with data this sparse, when you plot that data, so this, I'm going to minimize that project now. This project is talking us through the stages in that slide I mentioned from the FDA's paper. First of all, review your data. What does it look like? Well, in this case, it looks pretty sparse, and I don't know how I'm going to model that data, certainly as an individual. But we can apply the population tech to build a effective and representative model of that data, as I'll show in a moment. But certainly NCA is out of the window with this. Then here's another plot of the data where we can see, and let me just change that to see all the data at once. So we've got 10 APGAR scores here, and we can see different individuals have been grouped into different APGAR scores, and the healthier subjects have higher APGAR scores. They're more responsive. But again, each individual, very small amount of data. So how do I work on that? How do I build up a model? Well, let's have a look at this sub workflow I've built. We've got a base model. And in this base model, we've started with a IV 1 compartment model. And that means that when we look under the initial estimates tab, just build up in a moment. And I say I want to, this data ran for about 280 hours, okay? So you can see there's my loading dose and there's my maintenance dose. Let's look at that log, make it a bit more informative. It looks to be roughly in the right order for that individual. Let's see all of the individuals. So click overlay and we can see that's more or less where I want to be. If I want to change my clearance, I could say, okay, it's maybe 0.1, and you can see the data moves. So you can see this is the observed data, the circles and these lines are the predictions. Now, once you've analyzed that data, you can look through the results. So we can have a few things. We can have the theta. So this is the final parameters. How good are our estimates? How confident are we? And typically below 20% sounds pretty good. So I'm confident in my estimates. And we can look at the plots. So here's an overlay of all of the individuals. So those predictions are fitting each other well. You're only seeing a line where there's observed data, so that's why it doesn't look the same as this. And we can see how does that data look in terms of residual errors. So these are residual errors versus IVAR, the independent variable, which is time. And again, that's looking quite good. But let's get to covariate plots. And I don't have any covariate plots in this model yet, but let's see how we can add those for free. So I'm going to go to the parameters. Here's the structural parameters of my model. And what I want to go to is considering some covariate. So I click add covariate, but I'm going to choose this add from unused. And this is a nice feature. It just looks at your data, and we've got APGAR and weight, so we'll click an add. And those are now potentially covariates. I haven't put them into the model yet. If I was to click yes, then they get added to the model. Okay. I want to leave them out of the model, but just say I want to consider those as covariates. I can then click execute. I'm going to rerun this. I'll just take a few seconds. So what we're seeing here is something you do early on. You've got a structural model which seems good enough. Can I make that model better with covariates? Can I explain some of the variability? And by requesting those plots, what we get is these. So these are these differences from the population estimate would be at 0. And we can see this trend. So there's definitely a relationship between volume and weight, maybe volume and APGAR. It's got kind of a hockey stick relationship here, and possibly clearance and weight. So then what we do is we say, let's run a stepwise. So just like you do in Phoenix, you would do copy and paste. And in that copy, we take the run options from being a simple mode to a covariate search stepwise. And we can do that with various conditions by goodness of fit, which is one to look likelihood, or the Akaike information criteria, etc. And in the interest of time, I'm going to go to one I've prepared earlier. And this is the covariate search stepwise. And what it's doing is checking through each possible option. So on this first block, it's tried using weight on volume, weight on clearance, APGAR on volume, and APGAR on clearance. And then it's picked the one with the best improvement in the model. It's dropped from 1010 to 913. And we can see that weight has an effect on volume. And then it says, well, okay, if I know that weight has an effect on volume, does weight also have an effect on clearance? And it tries those different combinations, or APGAR on volume, and it brings in the next best model. And it keeps going, stepping through automatically. It just runs through. And eventually it says, I can't get any more covariates that are statistically appropriate. And then it double checks to see if it can remove any of them. It can't in this case. And that becomes the model to use. You can then simply copy and paste that to get the full results. And we're here. I'm not going to drill into everything, but you can just see that these plots have now leveled off. That trend we saw there has just become random noise. And the model is a better fit, and we have a better precision here in our analysis. Well, these have dropped lower than they were before. We have more confidence to predict in new populations. So this is how we build up our model. You might have seen a model comparer, but this shows a quick way to compare different models. And then, as I mentioned in the paper, we want to be able to easily assess those models for goodness, and we support those things like visual predictive check. These again are just a new option in the run mode. You just click, set up how you want to run that. We've got to bootstrap here as well, and ultimately we can perform additional simulations to check well, whatever we want to do. Here in this case, we're trying to demonstrate the variability that we might expect from our prediction and our observed noise. And that's what that overlay is doing there. lastly, I want to point out that we can take everything we learn in this Phoenix model language and extend that to using other tools, either the RSNLME. You may have seen some marketing around that already, some information about the new release last month of RSNLME. So this gives you an option for batching that up, but still using the same NLME engine from Certara, the same PML code you've learned and experienced in this sort of graphical mode. Alternatively, you can also use our code directly from Phoenix. So in here, you can see I've just got a little R script here, and when I execute that, it launches from Phoenix and brings the results back in, and there is some plot I might have done in R. So Phoenix is an engine, it's a workspace that you can use our own Phoenix's NLME engine to do everything that the guidance is asking for. But additionally, you've got some other things you want to use other tools for. You can build them in and package them up, and they just become part of your workflow. And that probably takes me to time. I know that was a very fast tour, but I just wanted to let other users who have seen Phoenix and using it for other features like NCA or IVRC, how they can take it further and really get more power from this platform. So at that point, Maria, I think I'll turn it over to questions.
Thank you, Simon, for this excellent presentation. We have one question so far, and please don't be shy to submit any other questions you may have. So the first question we have is, I see that you recently released 8.3.5. Can you talk a little bit more about that?
Okay, that's an excellent question. I was actually going to mention that if no one else had one. So yes, 8.35 was released in March, and this was essentially a maintenance release. It's not mandatory to update to, but there's some nice features I would definitely recommend if you are planning to do an update. It's a relatively small change control. What is that adding to you? We've given a feature that at every save, a small checksum is generated, and we check that everything has been saved correctly. The reason this is interesting is more and more people are using tools like OneDrive, and we sometimes find that as people are moving data back and forth across these synchronization tools, it can get confused, and so this checks that it is a valid save, and this would avoid any issues people might have seen with a corrupt project occasionally. And additionally, we have an auto backup. So every time you save your project, it will also take the prior save and relabel the extension to Phoenix Backup. Now, if for any reason that save became something you didn't want to use, so could have been a missave, could be something you didn't like, you can go back to that prior save, which has been automatically made for you, and that Phoenix Backup, rename it to Phoenix Project and reopen it, and you would have your prior save. If you're on 8.3.5, you don't have to do anything to switch on that auto backup. It's built into the software automatically as soon as you install 8.3.5. And remember, you can update for no extra cost. The license cost allows you to license subscription, gives you permission to go to the latest version at any time. The other thing we did here is we are always listening to our customers. We had just one customer experienced an issue with a very large NCA, maybe 600 profiles, and on that particular data set, they could occasionally get a Phoenix stopped working error. And it wasn't just due to the number of profiles, because we ran 800 profile data sets with no trouble. But we identified in the programming there was a small Stack Overflow issue. We fixed that, and to our delight, we've also significantly improved NCA performance for large data sets in general. So again, if you're running large bioequivalent studies, large numbers of profiles, 200 profiles plus, that might well be a good reason to update to 8.3.5. More esoteric differences is some people are setting up NCA and then saving them without having executed them, and very occasionally they would see those settings were lost. We've put in extra checks to ensure those are maintained. And lastly, we've improved the plotting output and calculations for some of our models where you've got larger models with multiple observation statements. So those are the key points for 8.3.5. Not a deal breaker if you don't upgrade, but definitely recommended for large NCA groups and anyone who's dealing with OneDrive, that sort of thing. Okay, any other questions, Marie?
No, Simons, that was it for this session. So thank you everyone for participating. We will now close this session, and as mentioned by Simon earlier, we will share the video with you later on this week. Thank you very much.
Thank you. And if someone needs the slides, feel free to contact us and I'll see what I can do.